novel delivery. Okay. Around novel deliveries of therapeutics to the brain, and in particular for the treatment of high-grade pediatric gliomas like DIPG. And so she's kindly offered to give us a talk this morning to kind of lay the groundwork and inform everyone about the type of work that she's doing. And the title of her talk is Multimodal Platinum 4 Drugs for the Treatment of DIPG. So, Nikki, if you can, I'll relinquish control, and if you can take it over, then you should be able to go through your talk.
Okay, Jeff, thank you. Okay, does that look okay?
Looks great, yeah.
Great. Okay, well, thank you so much for a chance to talk, and I'm really excited to find out more about what you're doing, and hopefully I can be of use to you as much as I'm hoping we can have sort of a two-way discussion, and I'm really excited about what you're doing. So I'll just start by telling you, I guess, a little bit about my background, just in case there are things which I've done which perhaps you may find of use at all. So, okay, next slide. Okay, so just at the top here, I'm a synthetic inorganic chemist, and I've done quite a lot of both organic and inorganic synthesis, and also looking at chemical sensing, so looking at basically the interaction of light with compounds primarily, and then I've become more interested in looking at also the use of ultrasound, and so how we can use ultrasound to deliver drugs. And so I'm going to tell you a little bit about some of the light activation that I've been looking at. So we developed these platinum azide compounds, which in the dark are completely non-toxic. But with irradiation, you can basically produce some very potent fragments within a cancer cell. So by cross-linking DNA with a platinum fragment, but also by producing radicals, azide radicals, hydroxy radicals. And so that's sort of, I guess, the backdrop to what I've started to look at here. Why I'm interested in DIPG particularly, obviously we all know it's the leading cause of death from brain cancer in children, and the fact that delivery really seems to be a key challenge for this particular tumour. And also the fact that combination therapy is likely to be a solution. And I'm very aware that I'm talking to people who know a lot more about DIPG than me, so I won't spend too long on this. But to move on, to give you a bit of a background, we all know that platinum compounds are widely used in chemotherapy, and these are platinum in the 2+ oxidation state. So I've shown the structures of the main platinum drugs here. Obviously, these have major side effects, and partly that's because they're very reactive. So what we're interested in doing is looking at making platinum 4 compounds. So you can see here that one of the advantages of having the platinum in the 4+ oxidation state is that you can have an additional 2 ligand positions on the platinum, and also the compound needs to be reduced in the cell before it becomes toxic. So this means that we can basically include multiple drugs within one compound, and we can also look at things like including fluorophores and targeted features. So this is, I guess, I wanted to give you an idea of what other people are doing, because most people within sort of inorganic medicinal inorganic chemistry looking at this aren't looking at a particular cancer. They're not looking at DIPG. So I have the advantage of basically really trying to read and understand the specific I guess drugs which we can look at incorporating, which will specifically be active against DIPG. So sort of some of the compounds which we're just starting with. So I've got a relatively small group. I've got 2 PhD students and 2 master's students at the moment. And what we're doing is we're just starting out by looking at compounds like indoximod, very simple ligands, and seeing if we can derivatize these onto platinum-4 compounds. So the ways in which we can do this is, you can see the carboxylic acid group there. We can use acid chlorides, we can use anhydrides. We generally need to be able to activate that position in some way in order for it to coordinate in the axial position of the platinum. And then coming on to delivery, you've already mentioned sort of blood-brain barrier permeability. So there are, I guess, 2 ways in which I'm looking at delivery. One is to look at liposomal loading. So obviously liposomes have been around for quite a long time, and there are quite a few liposomal drugs in clinical use. And the idea obviously is that we can, in theory, we can protect the drug from reactivity before it reaches the site. We can also include targeting features on the outside of the liposome. So liposomes themselves are not particularly responsive to ultrasound. So I'm working with Eleanor Stride in Oxford to look at using ultrasound-mediated drug delivery. Now, you can obviously use ultrasound to permeabilize the blood-brain barrier. So that's one option that we can look at. But at the moment, we're literally just looking at the release of the drug from the construct using ultrasound. And I hope that one of the advantages of this is that we can actually push the drug slightly further into the tumour than we would be able to do with just passive release. And I understand from talking to surgeons that DIPG, one of the problems is the pressure within the pons can be particularly high. So you're actually trying to administer a drug against a pressure gradient. So that's why one of the reasons I thought that ultrasound might be good for that as a delivery method. The second method that I'm interested in looking at, so this is, I've been talking with Stephen Gill, who is a surgeon in Bristol, and he does convection enhanced delivery of things like carboplatin. And this is essentially, I guess, cheating, because instead of trying to get the drug across the blood-brain barrier, you literally inject it directly into the brainstem. He's been very useful in sort of giving me suggestions and things that I can do. So that's, I guess, the background to where I'm at. Obviously, I've talked to Alex Bullock and I've talked to Paul Brennan about the things in which the advice that they have, sort of based on the things coming out of your network. If you had any thoughts on combinations of different inhibitors, I'd be very happy to talk to people about that. I've just listed a few that we're looking at the moment. One of the things which I haven't had a clear discussion with anybody on is the hypoxic state of DIPG. So it'd be really good to chat further with anybody who happens to know much about that. Alex and Paul suggested looking at M4K 2009. So I wondered if it might be possible to have a couple of MIGs just to look at combination cell tests, or if you're happy to share the synthetic route, we could probably look at synthesizing it at our end, if that would be acceptable, because then we could look at incorporating it into structures. These are the people I'm working with at the moment. I'm happy to answer any questions. Thank you.
Oh, that's great. Thank you. I think it's some exciting work. Maybe if you just go back to the question slide there.
Yeah.
I think we can certainly comment on the third question there. We'd be happy to provide you with both M4K 2009 and we probably have some intermediate around with the NH. It would be at least ready to be able to make that acid chloride. I don't know about David. Do you want to?
Yeah, I think we can probably we can give you 2009 and probably even make that acid chloride, and really we could have the discussion and we could actually make whatever you want. If it's simple and straightforward, we could certainly knock out a few things, whatever might be useful.
Yeah.
Okay, great, thank you.
Yeah. Yeah. And, you know, with respect to the hypoxic state of DIPG tumors, I don't know myself. You know, it's possible someone on the line may know, but certainly someone on the team will know. And with respect to combinations with ALK inhibitors, I know we're looking at EZH2 as a potential combination partner. I think PIK3 PI3 kinase is also one that I think we'd like to look at eventually. The ALK2 mutations tend to come along with PI3K mutations. There's PDGRF alpha that might also be an interesting thing to combine with. And, you know, I think the combination studies will, you know, all be very interesting. I think, you know, getting brain penetrant analogs for these different targets, though, remains a challenge. So I know that there are some brain penetrant HDACs that have been looked at that weren't that successful. But you know, it's something certainly that the team plans on looking at as we move forward in the project. I don't know if anyone on the line can comment about the hypoxia, the hypoxic state.
I guess that's a no. Okay, no worries.
Okay.
Yeah, that's fine.
Maybe we can follow up with the team about that later. So,
okay.
Hi Jeff, it's Alex. I think I talked to Chris Jones about that, and he thought there was hypoxic conditions for these tumours, but the extent I'm not sure about.
Okay.
Okay, great. Thanks, Alex. Okay, so maybe with that, Nikki, can you just maybe transfer control back to me and just stop sharing, and I'll
Perfect. Thank you very much. Okay.
All right, thank you. And I believe I have this one. So Jerome just got back from Japan, where I understood he and Alex were able to meet and talk a little bit. So that's great. Welcome back, Jerome.
Thank you. There was a great meeting, and Alex actually talked a little bit about M4K, and I think it got the interest of some people that were there as well, and you know, how they could contribute to this as well.
Yeah, no, I think that's great. You know, the more we can get the word out, the better. I think, you know, M4K 2009, I think now you'll see at the end of the presentation, I think we're almost ready to start sharing that for DIPG models around the world. So we'd like to make it as available as possible for whoever wants to study it. So.
Perfect. So I'll just update you on a few new things, and also a little bit of what was shown previously. So we tested a few of the most promising compounds in the DIPG cell lines. 3 of them here carry.
We don't have the slides up at the moment, please, Jeff.
Maybe I forgot to put. Sorry, I forget to share again.
Thanks.
Got it now?
Yeah, they're there now, thanks.
Okay.
Perfect. Yeah.
So most of you will have seen these data before, but we have 3 different cell lines here which carry ACVR1 mutation, all like K328, but they're different mutations. And one line that's DIPG6, which is ACVR1 wild type. And you see that the ACVR1 mutant cells, they respond basically to M4K, well, to all the compounds, but M4K2109, which is most interesting for us. So the next question was whether, you know, these compounds are able to inhibit the downstream signaling. So we looked at phospho-SMAD, and you can see they do. This was tested with around 1970 concentration. And to get a little more insight into that, I also did, this is new, these are new data. I did a dose response basically for M4K2109. In both the DIPG21 and the DIPG4 cells, the results were cleaner with the DIPG21, but they were consistent between the 2 cell lines. and basically you see that dose dependency inhibits phospho-SMAD1. In fact, this is not quantitative, but the IC50 with respect to phospho-SMAD seems to be lower than the IC50 in terms of affecting the DIPG cells growth or viability. There could be a bunch of reasons for that, including the fact that you might have to inhibit signaling down to a certain level before you see effects, right? So, and this would be also broadly consistent with also kind of like a lower IC50 that was measured when looking at ACVR1 activity itself with nanograph and things like that.
It's also consistent with the IC50s that Alex's lab has generated within the phospho-SMAD and the DIPG 007. The IC50 was around 70, 75 nanomolar or something like that.
So the
reading after 24 hours?
This is 24 hours, of inhibitors. We did some more experiments here to look at the expression of target genes, like BMP target genes, like the 192 and 93, and basically see that different compounds, including 21-9, suppress their expression in the ACVR1 mutated cells. That's just kind of like an additional readout. And then
I had a question.
Yeah.
Is the total expression different between the mutant cell lines and the wild type cell lines?
That's a good question. It's hard. I should go back and check if I can get kind of a quantitative sense of that. But it can be hard to compare. Like in terms of this is quantitative PCR, so you have to look at CP values. But even that's not like.
I mean, you would predict that those levels should be higher in the mutant.
You would,
right? I mean, that's
predict,
essentially the metric.
Yeah, you would predict, but then there's a lot of other things that can come into play, including chromosomal amplification and things like that. Other mechanisms. You see, for example, like ID2, which is a well-known BMP target gene, somehow it's not affected in the DIPG4 cells, but it's robustly affected in two other cell lines. So there's definitely some heterogeneity there as well. We looked at whether the compounds were inducing apoptosis to try to understand whether they had a cytostatic or cytotoxic effect. And in these experiments, we have this compound that we know exerts some cytotoxic effect. And it seems like mostly they don't induce apoptosis, but likely that at least what we see in vitro is related to growth inhibition rather than active cell killing.
The E6201, so, I mean, based on the fact that all of these other more selective kind of ALK2 inhibitors don't induce the apoptosis, do you think that maybe the apoptosis that we see with 6201 might be off target related?
I mean, or maybe on target. So the thing with 6201 is that it has several targets.
Right. Right.
It was not by design, but it was initially, I can remind you, it was initially designed as a MEK1 inhibitor, right? And it targets at least 3 or 4 times it has been characterized, which could be good, right? For one drug, right? Sometimes it's nice to have multiple targets engaged at the same time. So E6201 is actually a very interesting compound in that respect. And then, so a lot of the work that we're doing is trying to develop very good models of ACVR1 mutant tumors using both genetically engineered mouse model and xenograft. So far, our efforts with genetically engineered mouse model, they look promising, but they're complicated in terms of heterogeneity.
Sorry, Jerome. May I check with you in the previous slide with what method you used to quantify the percentage of apoptotic cells?
This is just stating for and 70.
And then with like how many days of treatment though? This is Zhongfu from Oxford, by the way.
I think this is 48 hours of treatment. I see something similar at 24 hours as well. I put it to 48 hours to see if I could see something else, more apoptosis or something coming up, especially because in the DIPG cell viability growth assay, it's 4 days of inhibitors. So,
because because over here, when I tried a similar experiment for 4 days, I do see like quite abundant percentage of like up to 70 or 80% of like apoptotic cells.
Okay, so with these compounds, and which cells were those?
But those were 007 from Engel, the one with R206H, yeah.
Yeah, so I haven't tested that cell line, although I'm supposed to get it soon. Yeah, that would be interesting to see if it's cell line specific or treatment protocol specific.
Yeah, it could be timing.
Yeah, it could be timing, yeah,
right?
exactly.
You know, because when I went back and looked at the data that was generated with the shRNA, at least on the DIPG 7007 line, it actually took 11 days for it to come down fully, right? Whereas at 4 days, you know, it was essentially a cytostatic effect.
Yeah. But, you know, even like what he was mentioning was that at 4 days, apparently there's some apoptosis in 007. But in my case, at least at 4 days, like if you look at these dose response curves, it does look like more cytostatic than cytotoxic, like, you know, the value that it reaches. So, but yeah, that's an interesting question to pursue further, like, you know, try and treat for longer.
Right.
See if there could be some cell line specific effects as well. So yeah, so going back to the in vivo models, so the most recent line I got from Stanford was this one, the DIPG36, and so they told me that it might be able to xenograft, so I tested it. So this is non-skid mice transplanted with 200,000 cells in the brainstem and postnatal day 2, using a setup like this. That's pretty easy to do. Thankfully, unlike the other cells I've tested so far, it seems like this one actually can xenograft and induce brain tumors. And in fact, this is quite aggressive. Maybe a little too much. So I might have to reduce the number of cells. Because if you think of moving this and testing a drug, the question is, when do you start the treatment with the drug? And I mean, you know, if you have to inject folks with all these 10 puff IV, like, I mean, you know, personally, I don't think I can do that. So, you know, maybe starting at weaning might be a possibility, but then if they start dying, you know, a week after, that might be too soon. And I have a second cohort of mice that's delayed by like a couple of weeks, and I mean, it certainly shows the same pattern already. They're coming down. So,
you have injected the same number of cells.
Same number of cells.
Yeah.
How big is the cohort?
This is 8 mice, and the second one is 8 mice as well. So.
No, where did you pick 200,000?
This is based on what other people have done in the literature. So people have injected something between 100,000 and 250,000 cells. So because, you know, I was interested mainly in first figuring out if the cell line can engraft, it makes sense to inject more cells. But you know, we can titrate that for sure. In fact, the question I got, so because I mentioned the route of administration, how do you envision that would be tested first? And like, what route of administration would we use initially?
I mean, this is a question that has come up relatively frequently. I mean, we're designing an oral drug, you know, so ideally it would be dosed orally, but, you know, for the proof of concept, IV, IP, hell, anything that gets the compound in, I think, will be acceptable for the proof of concept.
And that's something you need to map out. You need to map out an anti-MTD approach and IV-MTD for all of the different routes, depending on the data.
I mean, from a purely, you know, technical standpoint, like IP is the easiest.
Yes.
Like because the injections are simple and you know they're easy to inject, so you deliver maybe the drug more efficiently and more reproducibly. Galvin is technically a little more challenging, but and orally it's okay once you get it going, I guess.
Right.
We have recently done the MTD maximum tolerated dose orally, and it is working fine.
Yeah, I have some data to show about that, but the compound is well tolerated up to 100 mg per kg daily. So yeah.
with the lack of DIPG models, then so you're trying to get established tumors and then want to see if you can actually prevent growth. What about prophylactic? I mean, is that something that's feasible? Like, I mean,
well,
they injected that dosing and
yeah, we can think of like different ways of setting up the experiment. Of course, you know, it's not like patients won't be treated prophylactically with that drug.
I know. So with the lack of models.
I mean, but you know, yeah, it's an interesting question of with
yeah, there's a slightly lower bar, right?
Yeah.
You know, for the compounds, you know, and for the disease hypothesis. If you start dosing prophylactically, that's a possibility. Certainly something we could start with, but you know, the take rate was 100%. Essentially, all right, nice.
It seems so. Like there's only one mouse left in this cohort that hasn't got the brain tumor yet.
Did you open up at the end of the study?
Yeah, well, so unfortunately, most of them went down the way in Japan, but I got one yesterday. So, and then the second cohort, I'll be able to get more. And, you know, this another thing is like, this is a xenograft model. So yeah, the cells are in the brain, but you know, it's just really like the DIPG. Right? And they got interested in the brainstem, but it's almost certain that they're spreading elsewhere as well. You know, we could also put them under the skin, right? But it's not, anyway, I think the brain injection should be better, but it's still only a xenograft model.
Yeah.
So just keep that in mind. And I'm also getting the other xenographable line from Spain. So we have an MTA now with our lab, and they're supposed to send it, and so I'll have to grow it and establish it, but should have that going to test the drug as well here.
Great.
But I know maybe other groups that should be collaborating on that will be able to do that as well.
Yeah, no, I think, you know, like we said, you know, the goal is to make the compound available to as many labs as are willing to test it, so that we can get as much data as we can to try to understand more about, you know, the pharmacokinetics, the pharmacodynamics, and really just the behavior of the drug in these DIPG models. So. And others.
This is for me.
That's great.
So we do lack of any good models for DIPG. I mean, off the top, you can create a much different because it's probably more reflect pretty tell what you want to do. But to the question again, whether doing a sub, I mean, a lot of, you know, glioblastoma models, even though the tumors are in the brain, people sometimes inject them under the skin just to kind of figure out if a xenograft model is actually going to replicate what's in the prejudice. I mean, is that something you guys are?
Well, I mean, I think to me, like a xenograft under the skin, it's not that different than just growing the cell line just in vitro and putting the drug in, right? Like it's not. So I think the main interest of doing this is at least we're looking if the drug gets in the brain, right? Like and can therapeutic effect there. So I think that's the main question. It's the main answer that it's going to provide. It's whether the drug can achieve efficacy towards tumor cells that are growing in a microenvironment that's relevant. And also incorporating this whole issue about the blood-brain barrier and whether you can achieve sufficient exposure.
So, you know, I kind of agree. I think ideally it's going to be an experiment that's in the brain, but, you know, again, setting a lower bar. You know, running a xenograft to show that, you know, you can impact these cells and the growth, but I think it'd be in an in vivo setting would be okay as well. So, you know, again, we'll see what people around the world want to do with the molecule.
I think too, like, I mean, with regards to a lot of the intended combinations, I mean, with brain penetration, all of these getting in at different times, and when they go on a simpler model, just to kind of at least with a proof of principle, anticipate that this combination is worth testing. So anyway, that's something to think about.
Yeah, no, you know, I mean, the kind of lack of models is something that we did discuss, you know, and that we talked about at the the presentation to TPAC, and you know, I mean, they were in favor. That's why they were in favor of us moving into this FOP model. So something that is well validated, something that we understand, and you know, I think there just hasn't been a lot of certainly ALK2 compounds that have been tested in these models. So you know, I think we just don't know what to expect yet, and you know, there's more models being generated. So you know, I think we'll see as we move forward, but. The good thing is that we're starting to get compounds that are capable of being tested in these models. So, yeah. Okay. So let me just need to open Alex's presentation here. Where is it? I thought I saw it. Alex. All right. And there was Jung Fu as well. Where's Jung Fu?
Jeff, I think they're all in the same file.
Ah, they're all in the same file? Okay.
Yeah, right. The latest one, yeah.
Okay. All right, then Alex, this one?
Yeah. So we'll go through these very quickly, so hopefully we can not lose too much time. Ellie and Roz are going to present a couple of their new crystal structures. I thought I would just give 2 slides as an introduction on discussion around the sequence conservation versus the conformational diversity. So if you look on the left, previous crystallography of ALK1 and ALK2 at the ATP binding pocket, you can see structurally they're essentially identical, and then by sequence they're also virtually identical. And if you look at the analysis on the right, where you just kind of map out positions in the ATP binding pocket and then do a pseudo sequence alignment of ATP pocket residues across all the TGF-beta BMP receptor kinases, you can see there's very high sequence conservation. The 2 residue positions that are boxed would be the gatekeeper residue in the middle, where there's the shift from threonine to serine, and then on the first position, that's a hydrophobic, that sits above the solvent channel. So it's in the beta-1 strand, hydrophobic at the front. Okay. There are 1 or 2 other changes. In fact, there are other positions that aren't. Yeah, the 219 position, the second residue, is also interesting, and I've got some pictures of that on the other slide. Next slide. So when we've talked about conformational diversity, this relates in some sense to whether the protein's in active or inactive conformation. There's a couple of animations here. Jeff, if you could click the next.
Yeah.
So the ALK2 conformation we crystallize is an inactive conformation. Typically, this GS loop in yellow, which needs to be phosphorylated for activation, is buried at the back of the structure. This pushes the helix out at the back and in at the front. So that squeezes this site that should be phosphorylated, making it inaccessible. And that squeeze also pushes the ATP pocket in. And that means ATP can't bind, but obviously our kinase inhibitors do bind. This kind of trap structure is released by some of the mutations in DIPG and FOP, or at least made more dynamic or more flexible. Next. And that has the effect, if we compare active and inactive, that the C helix would then twist, and that's likely to be induced by phosphorylation of the GS loop when that's released, and that gives a change in the structure. So this is just an introduction to the next slide, if you want to move. Next. And again, sorry, I should have taken those out. So when I've looked, I've just overlaid a lot of the structures that we had, particularly a new structure of ALK6, this BMPR1B on the left, as well as, can you animate again, Jeff? Sorry.
Yeah.
Should have taken all those out. Sorry, there, yeah. So as well as the C helix movements, which we see between ALK2 and other type 2 receptors, we also see that in some ALK5 and ALK6 structures, while in ALK2, it seems more rigidly in inactive conformation. And then changes in the activation loop, which also squeeze the ATP pocket. And then in the P loop on the top left, Ellie's going to show you a structure where we have this extremely inactive conformation where the tyrosine comes down to bind the inhibitor. And all of these inactive conformations would allow our M4K series to bind but block ATP. But we see, I think, more conformational diversity in the different ALKs, perhaps than the sequence alone would allow. So I think some of the selectivity is due to sequence, and some of the selectivity is due to all of these different conformations we get. Next slide. So I'll hand over to Ellie, and she can talk about a structure, and then Roz is going to talk about another structures.
Hi, so I've just got a picture here of the pocket of ALK6 crystallized with M4K 2009, and on the left we've just got it on its own, so you can kind of see quite clearly what's going on there. We have one hydrogen bond with the hinge, as you'd expect for kind of an inhibitor like this, and what we're kind of not seeing quite so much is any water binding networks in the back. There are a couple of waters that you can see in the back of the pocket, but they're not directly bound to the ligand. This is kind of in contrast to the K02288 structure with ALK2 that I've shown overlaid on the right, where you do tend to see quite an extensive hydrogen bonding network at the back of the pocket. So that's quite interesting, and probably, I mean, I don't know, we haven't looked at BMPR1B in as much detail to see how essential that sort of water network is, but it does seem to be quite essential in ALK2, so it's quite interesting to see the differences there. You can also see kind of the key kind of conserved residues between sort of ALK6 and ALK2 are aligning quite closely, despite the fact that in the past the BMPR or the ALK6 structure has been in a different conformation. So we're seeing it more like ALK2 than we've seen before. Can we go on to the next slide?
And do you think that is because of the ligand?
Well, I mean, the only difference between the previous ALK6 structure and the current ALK6 structure is the ligand. So that's kind of the only sort of difference I can think of. I mean, it's possible it's just trapping it in that particular conformation, whether that's a transient thing that just happens to have been caught in this particular crystal form, or whether it's biologically relevant, we can't tell from just this static picture.
Okay.
So I've just kind of highlighted some of the differences here that Alex was pointing out before. So in blue, we've got the new ALK6 structure with M4K 2009. In orange, we've got the old ALK6 structure, so that was in the kind of the active conformation, whereas blue is the inactive. And then the green is the ALK2 structure that's in inactive conformation. So you can kind of see how the C alpha sort of helix there is more akin to the active structure, but when you look at the tyrosines in the middle panel there, it's very much more akin to the inactive structure. So it's much more like the ALK2 structure. So you can kind of, I've just highlighted various of the sort of structural features here. In particular, on the bottom there, you can see this loop has moved significantly up to match the ALK2 structure in our new BMP structure. And that's kind of exposing a potential new allosteric site that we've seen in sort of ALK2 before, and that Ross has been looking at in kind of XChem and things like that. So that's just kind of zooming in, looking at kind of the various features that are more or less or like other structures we've seen before. So that's my summary. I'm afraid it's not very much, but
that's great. Thanks. Yeah. Like I said, I think you know, we've got the structure, all of the structures here now available as well, including that ALK6 structure. So you know, we'll be able to start taking a closer look at some of these structural features as well. So that's great. Yeah.
Hi, hello there. So we've got two other structures about to with M4K 2117 and 2121 that I sent you a week or so ago. So this is just showing, well, I cleared out the waters in the latter images, but this shows the, oh sorry, it's tinkling. the histidine and the tyrus, the histidine backbone, where in the hinge region where you have that interaction that we've seen before, and then the trimethoxy coordinating with the water up at the top in the back, and that wasn't. So those are things that we normally see anyway. Although there are a lot of waters floating around, that's the main one that's involved. So if you go...
I guess the main difference in this one, I don't know how significant it is well, we previously had the piperidine at the back that you've changed, at the front solvent part has rotated round.
Yeah, that bit. So I've overlaid them later on, and you can see...
I guess that's the fact of removing that nitrogen, maybe.
Right.
Yeah.
So, yeah.
Change the geometry a little, whether that's stable or whether that's just captured in the crystal that way.
So on the next slide, so this is the 2121 structure that was around 2 angstrom. And again, I should have mentioned, in the other slide as well, of course, the lysine 235 is coordinating that water molecule that's coordinated with the trimethoxy. And then the hinge region is, again, the same if you see on the right-hand picture. So the density around the Solvent-side was a lot better, I must say, actually, than the M4K2117 structure, because actually that ring in the 2117 was kind of wobbling about a bit, I think. So the density was a little bit looser, but this one actually showed really, it was much more rigid as well, Solvent-side. But the other...
So it's a lot more rigid too, right? I mean, that may be why those things are typically more potent too, right?
Yeah. So then on the next slide, I've overlaid, so on the left there you see 2117 overlaid with 2121. You can see pretty much the main bulk of the ligand is the same, but at the end, in the solvent exposed side, yeah, the 2117 has twisted like that, it's rotated. And then if you, again, you see the same thing with the 4BGG structure with aldine 213844. So that, yeah, it was just to compare those two. And then I've done the 2121 and 4BGG on the next slide as well. So there, again, you can see, so the interactions are pretty much all as we've seen before, but just some movement of the ligand that's, around and bound.
So I'm sorry, so do you have a hypothesis as to what happens in the solvent exposed region?
Alexia, we had a difficulty hearing you there.
I'm just wondering, what do you think is happening in the solvent exposed region? Why do you see a lot of movement there?
I mean, I think it's just... Go ahead, Ross. Yeah.
There's not much, I mean, because it's exposed, there aren't really residues, or there is actually in the 2121, there is one water molecule there that's kind of coordinating, but I don't know how important that is. But yeah, that one. And yeah, I think it's just, it has more, it's possible for it to have more mobility. There aren't other residues around there that are coordinating.
I presume it does pick up a few van der Waals contacts and maybe the different groups.
Mm-hmm.
We'd have to look at the crystal packing as well, if there was crystal packing interaction. Do you know?
No, there was one in the asymmetric unit, and the packing was not, it wasn't around there. So.
I mean, Jeff, you probably view this part of the molecule still as just the part that we can optimize for PK, PD, brain penetrance, etc. Hug.
Yeah, no, that's actually a part of the molecule that we're working on, fairly extensively, staying close to M4K 2009, but making modifications there. So all of this information, I think, is really useful. I think that part of the molecule we think is probably what's responsible for the hERG inhibition that we see as well. So making changes there and trying to alter that part is I think, gonna lead to new and exciting molecules. So this is all really good information. And the, you know, like I said at the beginning, the 1.25 angstrom resolution of 2117 is really outstanding. So congratulations on that. That's really good. Yeah.
Yeah,
Okay.
it just meant there are a lot more waters in there than normal.
Well, that's good. That's good information to design compounds. We want to displace them.
For sure, okay. All
Okay.
right. Well, John
The
Fu.
final slide, John Fu's nanobret and nanobret assay.
Yeah, I'll be like really fast with the first few slides, and because this is just a very rough summary, and I'm sure David can explain much better later on. So I'm just having the 2009 as a kind of reference, like for the few slides, and of those compounds that have been like modified at the trimetoxy site, a few of them show like quite nice ALK2 activity as well as in red ALK5 selectivity, but most of them are like not really like that good. We can skip to the next slide.
Yeah.
And also there's some modification on the hinge finder that didn't really like result in like better compound, the compound that are better having better potency and selectivity than 2009. And can move to the next one. And it's with the Solvent-side, the analogs that are like very exciting. Some of them are really potent as well as really, really selective. So yeah, there are quite a few of them, and it seems like quite promising, as well as the options that are like possible are like quite pretty much open, I think.
Yeah, no, I think that report was very interesting. We are starting to get some really good selectivity against ALK5, which is something that we've been trying to get since we started. So, you know, that's some great information, and we're happy that ALK5 assay is back up and running. And you know,
yeah, I've been having some technical difficulties for quite some time, but I'm happy.
Definitely some useful information there. So. Yeah.
And among them, the compound that I've tested, I noticed there are two that are having the linkers that were meant for subsequent, like linking to 404 for the ALK5 nanobread assays. Unfortunately for those, right, I haven't included them in the slides. The IC50 for ALK5 increased by 15-fold or even more, so like might not be extremely encouraging at this stage, I think. Like at least for those two, 2144 and 2147, I think. Yeah, those.
Yeah, I have a quick comment in my presentation on that, John.
Yeah, that's great. Yeah, we can move on to the even the next slides. Yeah, can move on. Yeah. So this is one interesting thing that I've been testing out as well in the meantime. Which is looking at the binding kinetics of the M4K compounds in the cells, and this is done by nanobread. So I saturated the ALK2 by incubation with 20 times the IC50 of different compounds, and after that after 2 hours I wash out the compounds by washing in like fresh medium, and afterwards I incubated in saturating concentration of the nanobread tracer, and follow how the tracer start binding, and you start seeing bread signal through time. And from this assay, it seems that M4K3007 is having really stable binding to the ALK2 receptors, and in comparison, 2009 is not too bad, but like 2127 and 2141 seems to be better. But this is
worse, right?
The residence times are like in terms of like the slope, like how inclined the.
Oh, sorry, right, yeah, okay. Okay.
Yeah, but I'm still in the process of sorting out whether, because the assay is complicated by the fact that this is done in the cells, so the compound could also be trapped within the cells, and therefore after the washout experiment, there will be rebinding of the of the compound within the cells back to the ALK2 receptors. So I'm still in the process of like eliminating the possibility that that's the case.
Yeah, like a proportional to protein binding or something like that might keep it in the cell. Right?
Yeah, although I'm not sure like how useful this information would be for us. Yeah, because it does take in consideration that the diffusion, like the intake of the compound into the cells, and also the efflux of the compound out of the cells as well, so the diffusion as well. So, and possibly the more hydrophobic compound might be trapped in the cells longer. So, but potentially this might be helpful for us to evaluate compounds with like similar potency.
Yeah, it's interesting. I mean, we usually get our kind of residence times from like SPR experiments or something like that. But you know, outside of this, so you remove all of those complications with the cell penetration, and you know. But yeah, I think you know, there's definitely some interesting data there. It'll be interesting to see whether or not we can correlate that with anything as we move forward. Yeah.
Thanks. And next slide. And in the last two slides, I'm just showing some really preliminary data as well. And these are using cell titer glow, where I'm measuring the ATP content of the cells. And I've done some experiment with 2009 with 4 days of treatment, and at that time point, it seems the GI50 is not that great. Maybe we can move on to the next slide.
This is with the DIPG 007?
Yep, with the R206H mutation.
Yeah, I mean those IC50s are very similar to what I've seen in the Stanford cell lines that I've tested here. So
yeah, 4 days. Yeah.
And subsequently, when Liz, the PhD student, when she tried a similar experiment, but with only a single time point, where she tested like different compound, but for 7 days, and at a single 1 micromolar concentration, she noticed that M4K 2009, for example, there's over 80, like almost 90% decrease in the viability in terms of the ATP. So from that, I'm thinking that the efficacy of the compounds, it probably, the actual efficacy is probably higher, but I just need to check the different time, treatment time for the cells.
So, yeah, I mean, I find it a little strange that all of our compounds don't behave at least fairly similarly in this assay. Right? I mean, they all inhibit ALK. They all have pretty similar EC50s. So the fact M4K 2009 almost seems to be the outlier in this experiment. Am I reading it right?
That's true, Jeff, but they're all just at 1 micromolar. So if you raised it to 2 micromolar, then maybe they would all look a bit more similar. We don't know.
That's possible, yeah.
This is a single concentration point, yeah. I'll be doing.
I just want to comment on this assay as well. So I see that you label as relative viability. This is just a point I wanted to bring up because this assay actually is exactly the same as I'm using. It's measuring ATP consumption, so it's basically measuring metabolic activity, which could be a combination of viability and the number of cells that you have merely. Yeah. So it's just something to keep in mind.
For sure. For the subsequent assay, I'll be quantifying more parameters.
And do you change the media or anything in this assay over 7 days?
Not, but very low cell amount was either in the beginning, so that they did not starve.
Yeah, I would have a similar question. For the long experiment, do you decrease the density? You know, for the long experiment, or is the same density and keep the media the same, or do you do any splitting?
Basically, the medium and the compound were just added in the beginning and never refreshed throughout the 7 days.
Have you thought about doing the like a colony assays or encourage independent growth assays?
Liz, the PhD student Liz, she had been testing out some of these assays.
Yeah, because generally, you know, the agar outgrowth assays seem to be sort of a reasonably good predictor of behavior, where you get sort of benefit of seeing both the effect of proliferation and cell death, and how that contributes.
I don't know if anybody has tried these cell lines in an agar assay, actually. They grow as they all grow very differently. The different cell lines, like some of them grow as neurosphere, some of them as air, some of them the mixture. And I actually tested out in 2D and 3D. So some cell lines, depending on the plastic scheme, they can grow them into section or grow them attached. I don't know if anybody has tested in something like soft agar or colony forming.
Yeah, I don't think I've seen
that. Does that correlate with aggressiveness of the cancer?
I don't know. It's a good question.
Yeah, that's hard to say.
Yeah, I mean.
Okay, yeah, no, I definitely think that some of these longer-term assays are something that we do want to pursue to see whether or not we are affecting proliferation in a longer time frame experiment. I think this is leading in the direction that maybe we will see more enhanced anti-proliferative effects of our compounds, but I think we need to think carefully about how we want to do those experiments. And I mean, we have some experience here at OICR in running longer-term proliferation assays. We've seen that ATP light assays can be somewhat misleading in longer-term assays because cells don't usually like to stay in their own crap for very long, so you do need to split them.
These cell lines in general, we keep them in defined medium for 7 days, and then we change the medium, so that's pretty much the limit. Basically,
right, yeah, and then we've done splitting as well, right? Assays with splitting and redosing with compound. That reminds me that one of the comments and one of the suggestions from the TPAC committee was because we are starting to look at some of these longer term studies, was to look at the stability of our compounds in these assays as well, just to make sure that they're sticking around. I don't think that it's an issue, but it'll be good to get data just to demonstrate that. So just a simple HPLC experiment, I think, demonstrating that the compound's still around at the same concentration.
It can be done very easily at time zero, like just get a small amount of media at time zero and up to 7 days, you can just run HPLC.
Right. Yeah. So that's something maybe we should do, but let's keep thinking along these longer term cell line or longer term cell proliferation assays, and it'll be interesting to see what we come up with there. So that's great, John. Yeah.
That's all the results from Oxford, Jeff.
That's great, Alex. Thanks. Thanks again. All right. Who's next? Sue, I believe. Sue, do you want to take control?
No, it's all right. You can run it from there.
Okay. All right.
Okay. So just a general update on progress. So since the last meeting in September, we have made and I've sent to screen or are about to send to screen about 36 new compounds. And part of this was as a result of our global week of service in early September. So this was some of the output. So there's been further analogues in the pyridine series as well as some hybrids of the blueprint and pyridine. So we're looking to, so we're looking to try and now identify a second series. So to that end, we're again looking at more hybrids of blueprint with other ALK2 inhibitors, as well as what we might be able to do around the blueprint compound M4K3007 that might improve its properties. So we carried out, just a reminder, we carried out a PK study at our CRL sites on both 2009 and 3007. So if you go on to the next slide. So I'm going to pass over to Dawn, who hopefully is on the line to talk through some of the results that we've got.
Hi everyone, yes, Sue, I'm here. So here's the PK data for M4K 2009 with the Farmer data on the left and the Charles River data on the right. There are some differences in study design. For example, the IV dose level for the Charles River studies is 1 mg per kg and the Farmer studies 2 mg per kg. In the table, we've got a comparison of some of the key PK parameters from the two studies. I'm just touching on the oral data initially. So the plasma concentrations and AUCs, etc., are a bit lower in the study performed at Charles River compared to Farmeron. I think they're within sort of limits of what you would expect for biological variation. And I think also when you compare that to the oral exposures obtained in the dose escalation studies, they are a bit lower, but they're thereabouts of what has been seen in the other studies. What is notable in the studies is that the plasma concentrations for the IV legs of the studies, we do seem to have lower concentrations in the Charles River study compared to the Farmeron study, even accounting for the difference in the dose levels. And that results in us seeing higher clearance and a higher volume distribution compared to the data generated at Farmeron. What I only realized myself recently is that the Farmeron data is done in the SCID mice. So there may be a contribution, or that may be contributing to the difference in the PK that we're seeing, because with the immune deficiency, whether they behave exactly in the same way pharmacokinetically, I don't know. So that might be a cause, a potential cause of that variability.
I have a quick comment about that. Like, was the dosing volume and the dosing formulation were similar or same for them?
The formulations are the same for IV. They're similar for oral. I think we just took the tween out for the oral dose. I'll need to check the records, but I believe the dose volumes were similar in both studies, but I will check and confirm.
Well, the Charles River, IV dosing does look a little bit unusual, because you see the 1 hour time point going up in the IV arm. Usually, I've rarely seen anything going up after 1 hour.
Yes, so this I think is because we're taking average data. So what we have got is a composite study design where we were using 3 mice in the study and taking 4 samples from each of those mice. So every second time point is from a different mouse, rather than serial bleeding throughout the time course from individual mice. So I think that is perhaps a contribution to the increase, rather than it being pharmacokinetic.
Yeah, so we do the same thing. I mean, we do a serial bleeding, with multiple groups, but even then, it's a log scale. It's quite rare. I mean, your concentrations are very low to start with, and then that point is not usual. And then I'm pretty sure we couldn't get a good R-square for the fitting because the last few points are quite scattered.
Yes, that is why there is no half-life reported, because the values it was coming up with wouldn't have been appropriate for the time course of this study. Yeah, that's correct.
Yeah, no, I mean, I think the oral exposure is still, as you say, within experimental error. I think, it does look like we had some solubility issues maybe in the Charles River study. I noticed that it was dosed as a suspension at Charles River, and it was a solution at Farmeron. And sometimes when we make our formulation, usually we dissolve our compound in DMSO first, and then add the water last, essentially, right? So that might account for some of the differences we're seeing as well, the solubility issue. So, but you know, the oral exposure is still pretty good. I think that, you know, one of the things that I liked out of the Charles River study was that we did see a 1-to-1 ratio of plasma to brain in that study. So I think, you know, the fact that we're getting good exposure in the brain with this compound, it still comes out of this study as well. And you know, if the plasma exposure is a little bit lower, but the brain exposure is a little bit higher, I'm perfectly fine with that. So, you know, I think I agree with Ahmed. I think the IV leg in this study is a little odd, and I tend to, you know, err towards the Farmer study for our IV parameters. But you know, I think it's something that, you know, we should look at moving forward if we do any additional studies at Charles Rivers to try to understand why we're seeing this. I noticed as well, anyway, I guess you'll talk about 3007 on the next slide.
Yeah, we can move to that now if you wish.
Dawn, did you have any major concerns about the results that we had?
It is, I agree with everything that's been said. It is an unusual looking profile. It is lower than we were expecting based on the Farmer data. So there may be some practical things in there that are contributing to that. It's difficult to pin it down exactly to what it would be. And one of the contributing factors to the variability would be perhaps the difference in the strains of mice or the age of the mice. For example, but I think, you know, the take-home message, what is in both studies, we do seem to be getting good oral bioavailability.
Yeah.
And in both studies, it's greater than 100%. So I think it's suggesting there's some saturation of something somewhere. Don't really know what yet, and I think we'd need more data as the project moves forward to try and understand what's contributing to that greater than 100 bioavailability.
Yeah, but it does highlight the importance of, you know, getting additional sets of data for important compounds as we move through the process. So,
do we have that? Have we done that PK?
We haven't done rat PK yet, no. But it'd be interesting. Maybe we will.
Yeah, because anyway, you know, sometimes I look at some compounds in development from peptides, and you know, they'll do all of the mouse studies, but the reported PKs in that. It's probably for a reason. The dosage compound are only mouse for the efficacy, but they never report PK data. They report all of the PK in that. It's just easier to handle. Dosing is more convenient. I mean, so maybe to get real PK parameters, maybe that might be a species to just try to please because they're not.
Yeah, I'll take note of that, and I think we should be able to run one of those. Yeah, I did notice as well that there were some differences in the dosing volumes as well. I think, you know, generally Charles River dosed half the volume that Farmer did in both IV and the PO arm. I think so that could account. So there's several differences that, you know, I think the results are close enough, at least in the oral leg. We can safely assume that we can move forward with 2009. So,
this slide covers the PK data for 3007 performed here, and I think it shows that we've compared to the 2009 study performed here as well. We have similar clearance and volume distribution between the two compounds, and the good and high oral bioavailability has been retained. We do have a lower brain to plasma ratio for this compound, suggesting that the penetration to the brain may be less favorable than 2009. Whether this is related to permeability and efflux restricting the penetration to the brain, that will be useful to get some CATO or M4K data to see if that is a contributing factor to the lower brain distribution for this compound.
Yeah, we can put M4K3007 into our KCO2 assay here, and we'll get that data. I'm actually surprised we don't have it, but it's possible. But yeah, we'll make sure to get that tested. Yeah.
Sorry, Jeff.
Okay.
I couldn't see it in any of the data that you passed on. So.
Right. Yeah. No. I looked this morning in our database, and we don't have it. It's not in the database, so we'll get the data for sure. Yeah. But I think it's interesting though that this compound does have good bioavailability. The fact that it doesn't penetrate the brain is okay, but I mean we are getting about 20% in, which isn't terrible as a starting point. So as a backup series, this I think is interesting. Yeah, and I know you have some plans around that. So yeah.
So I'll come back to me now. So just looking at the progress, as you can see from this slide, we've been looking at a broad range of heteroaromatic replacements for the trimethoxyphenol, trying to pick up the same sort of interactions, but perhaps without the potential issues that you might have with the trimethoxyphenol down the line. Obviously, I can't comment on quite a few of them because we're waiting for assay results on a batch of compounds. Just to point out, I think at the last meeting, Sylvia discussed the fragment hopping approach, and the last three compounds on this slide, the bicyclic analogues, there were compounds that had been identified from that fragment hopping exercise. So it'd be interesting to see whether any of those show any interest.
Absolutely, yeah.
So there's one or two more compounds that are just being finalized, synthesis. But for the moment, I think because we've got quite a range of different structural types, we'll sort of hold on that. We've made some of them where the R group is methyl, and some where the R group is H, because I know in some of your areas, in some of the compounds that you've made, you've actually seen differences, and this may be due to the way that group orientates in the pocket. So until we've got some more results, we're sort of holding on any more compounds in that area. Should we move on to the next slide then? So the compounds on this slide were some initial attempts to overlay. So you can see on the right-hand side an overlay of the sort of piperazine compounds, which are designed as mimics of the blueprint compound. So we've taken the pyridine series and put on these piperazines or pyrrolidines, and we're waiting for results on the majority of these compounds. So these could be interesting.
Yeah.
Next slide, please. So I had a few thoughts about why the blueprint compound may have poor brain penetration. Now, it may just be an efflux, but it could also be due to the fact that it's going to be very highly ionized. It has a pKa of 9.9. So I was looking at what part of the molecule was contributing to that high pKa, and have proposed a few compounds. Okay, the middle one probably doesn't quite address the reduction in the pKa, but the one on the left and the right, potentially could give you a similar compound but with improved brain penetration. So move on to the next slide, please. So we also have started to look at what else could we do around the blueprint compound. So as you said, it's not perfect, but it's very potent. And I think the results that we saw in the cell viability were quite interesting. It seems to be behaving differently from the pyridine series. So I think that I was looking back through the patent, the blueprint patent, and to see what was actually covered. There's very little covered where they modify the core. So it looks like we could do some various changes to that core. And what we've looked at initially is what is synthetically relatively easy to make. Some of these cores here, the intermediates are commercially available, and some are just one or two relatively straightforward steps. Obviously, the cores on the right-hand side is substitution on the pyrrolopyridazine moiety, where that may again influence the pKa. These are described, these synthetic approaches to some of these substituted cores are described in the literature, so not for these compounds, but for other types of compounds. So these are some other scaffolds that we think we can look at.
I wonder, you know, I like the idea of exploring the blueprint compound as a backup series. I don't know that we need to build in IP at their intellectual property or move away from their patent. That, you know, at this point, I think, you know, if we could identify a compound from their series that has good brain penetration, at least we would get the proof of concept and we could move forward with that. So I think, you know, these are great ideas for kind of circumventing the patent if we have to, but, you know, again, there's not a lot of money to be made in this indication, so I think, you know, we'd be able to approach Blueprint and maybe in-license their compound or something, one that we identify that does have brain penetration. So I like the idea of exploring a scaffold. I don't know about, you know, maybe these might be something we could consider further down the line once we demonstrate that we can get it across the blood-brain barrier. Yeah.
Part of the reason for some of these was there because the scaffold, the blueprint scaffold, is actually quite complex to make. Some of these are relatively simpler, so that was our sort of starting thoughts. But I think we can explore both. Look at some compounds within that where we can perhaps reduce the pKa of the molecule, as well as some things that fall outside if they're synthetically readily available.
I mean, the bigger question is, I mean, usually for brain penetration, the smaller the molecule, the better. So the question is that enough ligand efficiency? Is that an exercise that could do? Can you shrink that scaffold down to a meaningful molecular weight for brain penetration? I mean, so whether that's a worthwhile exercise as opposed to maintaining that high molecular weight for CNS penetration?
Certainly, yeah, the molecular weight is high on this, and even just taking off, so if you look at the previous slide, you know, you can see in the right-hand side, if you take off the ethoxy, for example, you reduce the molecular weight. So that was some of the thinking. So the two things that seem to be are a high molecular weight and pKa and high pKa. So that was the thinking behind those.
Good. Okay, good. Anyway, we should probably move on because we are rapidly. Oh, okay. That was it. That's great, Sue. Thanks as always. David. Where are you? No.
Second one, third one? No, that's one.
All right.
Okay, so in the interest of time, I'll kind of highlight the critical things this week. One of the critical things that we've had on the menu for a while was to look at some of the other active compounds in the series to see what they look like against the ALK2 mutants. And so we've done that now, and so we can, I think with confidence, say that if we have activity on ALK2, we're going to have to be essentially equivalent on the ALK2 mutants that we're looking at, and this series bears that out. So I think we've established that quite firmly, so probably no need to test any more compounds against these mutants in the time being. Here's a closer look at 2009, kind of a little bit more tabulated look at its activity across the isoforms that we're testing, the 3 mutants, and then all the isoforms up to ALK6. Here's the activity in nanomolar, so you can see that we're actually quite selective. Here's the selectivity index in the table below. You can see that we're quite selective against ALK4 and 5, which is what we want. We're clean against those to limit potential tox issues. And we're equipotent on the ALK2 mutants. We're a little bit of a window against ALK3, and also we're essentially equipotent on the ALK6. But again, it's good that we're seeing a window versus the 2 isoforms that we're really interested in not hitting to any degree. This is a little bit of a recap of what John Fu showed. He's been running the ALK2 nanobread versus the ALK5 luciferase to check the cell-based selectivity. So it's a little bit of an apples and oranges, but we're still starting to get an idea of what compounds are not hitting ALK5 in the cell-based environment and the selectivity versus ALK2. And so these are some of the compounds distilled from that set. These have a biochemical ALK5 selectivity over ALK2 of more than 100-fold, the cell-based greater than 80-fold, and the ALK2 nanobread values under 100. So you can see we have some compounds which nicely meet these criteria. So we're going to be starting to kind of look at these in a more fine-grained manner and take some of them forward through profiling. These are the more potent ones by the ALK2 nanobread. So these are kind of in the 50 nanomolar subregion. And then we have also a series which kind of drift a little bit higher, above 50 nanomolar. So again, this is kind of, again, the second tier of the best compounds. With the exception of this one right here, which is the inclusion of the nitrile and this unsaturated piperidine, really nice selectivity, good ALK2 nanobread potency, and good biochemical potency. Just a quick comment on substitution of the trimethoxy with something else. So we've tried a number of compounds. We now have this inside methyl fluorodimethoxy compound. And while we do have some reasonable activity in the ALK2 nanobread, the selectivity is not optimal, but it's not terrible either. But that means that this group is potentially something we can look at if we're interested going forward as an alternative to trimethoxy. The other alternative is the methoxyamide. We now have the matching inside methyl for that compound. What's interesting here is that we have pretty good ALK2 biochemical potency, reasonable in the cell-based, but interestingly enough, the addition of this inside methyl has not dramatically increased the cell-based potency, cell-based selectivity over ALK5. So this is an issue that we can also potentially look at going forward. And we have a new member of our team, Diba Ensign, who's an MSc candidate here at OICR, to be looking at some of the amide modifications to see if we can address the issues which are inherent. Because this is quite a nice group, which is giving us some very nice potency, so it would be a shame to throw it away at this point. See if we can develop it further. We were also looking at the piperidine core modifications. Here are some, the southern solvent exposed modifications. These are some that I showed last time. One recent one which you've seen is this sulfonamide here, and this is an interesting result. Again, we're showing some pretty good cell-based activity, and we're showing a very nice window here over ALK5. And this is a compound which we might be looking at, for example, the hERG issue that we're having. This may be a way to address that. So again, we might take this compound forward into the hERG profiling. And because this result is quite significant, we have a couple other team members which are newly on board, Dimitri and So Young, and they're going to be doing some more extensive explorations around this piperidine group or piperidine modifications, et cetera, to see if we can address not only the hERG, but also for the issues of selectivity and whatnot. So hopefully they'll be reporting more of those compounds in the meetings to come. Here's a couple that have already been made by So Young. We had to get the SAR completed for the inside methyl morpholines. In this case, unfortunately, we're seeing that the ALK2 nanobread is drifting higher, so this may not be optimal for us. And we also have the N-acylated compound. Again, we're seeing the ALK2 nanobread drift higher, but again, we need more kind of fulsome exploration around this solvent-exposed portion of the structure.
You should make this cyano that comes out because maybe somebody different.
Right, exactly. And that's the intent. Yeah, we want to. Yeah.
Maybe less soluble, but to make it very, very potent, and so actually it's going to be okay. I think they're going to be more permeable than the reason.
Yeah, we're interested in finding solubilizing groups on the amide as well on the lower side, to try to recapture some solubility. But right, so yeah, I think that there's a bunch of interesting compounds that are going to come out of that. And it seems relatively well tolerated. It's the solvent.
The other interesting result was looking at the benzoic position and substituents, a la the blueprint compound. And I think the most interesting outcome here is the old methoxy. We're potent, very potent in the nanobread, potent in biochemical, and we also have a reasonable selectivity window already. This is minus the inside methyl, and that compound is being made right now. So we'll be able to complete that set, and this might be an interesting motif to carry forward. The other one is not so much, with the exception of this unsaturation in the piperidines, which is also showing some very nice selectivity. and we're incorporating that into a number of compounds we want to look at further. Here's a couple of combinations of the unsaturated piperidine with the inside methyl and nitrile, and you can see very nice selectivity over ALK5 and the cell-based environment, and also very nice ALK2 potencies by nanograms. So this is definitely an interesting motif to see if we can take further. These oxopipridines didn't really kind of, they are potent in the biochemical, but not very nice selectivity over ALK5 in the cell-based environment. So we wanted to see if we could modify them, get rid of the oxygen, start looking more of these piperidines and pyrrolidines. And so there's a couple of interesting examples here which we could potentially build out to modulate the overall basicity of the molecule and bring down the log P. So again, this is a work in progress. Here's just to highlight a couple of the tetrahydroisoquinoline cores. Here's an example of where introduction of the inside methyl group did not, it did increase the selectivity for ALK5 in the cell-based environment, but perhaps not as dramatically as we would have wanted it to. Although the compounds in the first case without the methyl, interestingly enough, are quite active, but the introduction of methyl promotes the activity a little bit. So some more exploration to be done around this tetrahydroisoquinoline portion. Combining that tetrahydroisoquinoline with the different warheads, unfortunately in this case not very satisfactory because we lose the selectivity over ALK5. Although the incorporation of the nitrile and the isoquinoline together does again open up a very nice window and keeps very nicely potent compounds. So 2143 is something we'll be looking at going forward. It's one of the more interesting compounds. This series of substitutions on this inside, or sorry, the central arrow ring haven't really yielded anything dramatically interesting, with perhaps the exception of this orthochloro compound, which again, quite potent, but again, something to be desired in terms of the selectivity. So perhaps making the inside methyl or nitrile is something to complete the set. And in terms of the inside position substitution, we've done the nitrile. We've now run the series here with the CF3 and the fluoro. The chloro has been made. We're still waiting for the result here, and we're expecting it to be probably equipotent to the inside methyl. So we'll see what happens there. The interesting thing with the inside nitrile here is that it did appear not to confer a great degree of selectivity in the cell-based for ALK5. But interestingly enough, when we look at the very closely related compound, the unsaturated piperidine, we do see the nitrile, in fact, conferring quite a bit of selectivity in the cell-based environment too. So I think some more examples of the nitrile are in order to see if that pattern holds. This is just a checklist, which I showed you before, of the criteria that we're meeting for kind of overall desirable compound. The one liability that's still persisting, if it is in fact a liability to any great extent, is the activity on hERG. And so to that degree, we've profiled a number of compounds, a variety of chemotypes in terms of the southern portion of the molecule. And what we're seeing here is essentially uniform activity against hERG, somewhere in the kind of 3 to 10 micromolar region. And so this is something that we're going to be looking at, trying to see if we can tune out by modifications of the southern portion of the molecule. And so efforts are ongoing with the other students that I mentioned. Lastly, just in terms of taking the chemistry in a little bit of a different direction, we know that this, the position, this 4 positions does tolerate a methyl group. So one of the things I've been working on lately is to see if we can do a core modification whereby we swing that entire southern moiety to that position. And to see if we can actually retain the activity. And these compounds, interestingly, have docked very well. So it'll be interesting to see what the outcome is like in the actual assay, and to see if we can still retain the benefits of the selectivity and have a little bit of a different orientation of the core. And lastly, I just want to comment on the efforts for developing the ALK5 nanograph tracer. As Jiangfu commented, the cell-based activity for these 2 compounds, which are the precursors, the linker and Bach compounds, they were around 1 to 2 micromolar in the cell base. And we had a little bit of an email discussion with Carol at UNC, who's going to be attaching the DIPG fragment onto these guys. And she thinks it's still worth pursuing. And I believe that Alex and the guys at Promega suggested that, we have these linker compounds made, so let's just attach the tracer and see what happens. We may have something that may not be suitable for ALK5 and may be suitable for ALK1 or secondary ALK. So that's where it stands right now in terms of the YCR chemistry.
Krista, what's what's your criteria for selectivity? Is it the regards to the biochemical or cell-based potent? What fold selectivity are you looking for?
So our criteria is greater than 10 micromolar or 30-fold higher or better than the top concentration that we'll hit, essentially. But again, I mean, this is a binding assay, the data that we have, right? So I'm not sure how much.
So that's not channel, that's not data,
It's not, yeah.
Patch
It's just a binding data.
It's just a
binding assay, yeah.
Oh, it's not patch
Yeah, it's not patch no. So that would be next, and then dog telemetry would be, right? So I think that there's a couple of additional experiments that we can do to really see how much of a liability it is. I think the potency that we're at in this assay is actually pretty reasonable for these compounds, given that they do kind of have that herb pharmacophore, right? So
this is not, I mean, if it's not patch clamp, I mean, you should find out what the patch clamp is for some of the, the might actually be better.
Yeah. Yeah. No, that's in the plan. You know, again, trying to move it forward without spending a fortune too. Okay, where am I? All right, so again, typical that I said that we would have plenty of time for discussion. Well, we've had the discussion throughout, so I'll be able to kind of go through my section pretty quick. The updated up-to project flow scheme that we showed is kind of shown here. The only difference is that we alternate in terms of where we put these phospho-SMAD and target engagement assays in the cascade. In the proliferation assays, again, the data that we're getting there, we're not sure exactly what to make out of it yet. Do we need an IC90 to actually impact proliferation? Is it related to the cell lines? Is it related to duration of the experiment? So there's still some work and some optimization going into trying to figure out how to best to profile our compounds for their anti-proliferative effect. And you know, we'll see some data hopefully shortly with that. Then the compounds move in vivo. We've looked at pharmacokinetics. We've looked at 10 compounds externally so far, as well as a number internally in our rapid screen PK. We've looked at 5 compounds, now 6 actually, with the brain to plasma ratio. We profile them against other kinases. I'm actually looking at RIPK2 and some of these other kinases that are available in RBC. And then David showed nicely that, you know, our wild type ALK2 data correlates with the ALK2 mutant data, so we only generate that data on compounds of key importance. And then moving into advanced in vitro ADME, plasma protein binding, brain tissue binding, CYP inhibition, looking at some initial read on safety pharmacology, the HERB, the AIMS, the SERA panel, and then more extensive kinome profiling. And then looking at, going back in vivo, looking at dose escalation studies and a maximum tolerated dose study. I'll show you some data on M4K2117 that we just generated there. And then we will move the compound into efficacy studies, starting with an FOP model that we know is validated, that we know is dependent on ALK2, and will respond to a good ALK2 inhibitor. So we hope to be able to inform a DIPG study based on that data. So moving in now to advanced profiling of M4K2009. Plasma protein binding was 91%, the brain tissue binding was 99%. Again, this is something that maybe we can try to improve as we move forward, and we've had some discussions about that and how relevant that will be. But it is something that we're cognizant of, and we'd like to try to improve if we can. CYP inhibition, we didn't inhibit any of the 6 major isoforms at Charles River. HERD, as we said, is about 1/4 micromolar. It was negative in the AIMS assay and hit only 10 out of 374 kinases at 1 micromolar, and none of the kinases that it hit really represent any major concern. It was also tested, I'll just move on. The SERA panel, the safety screen 44, we did hit a couple of receptors, in particular the cannabinoid CB1 receptor, which was inhibited at 97% at 10 micromolar. This is probably something that, you know, we would want to dial out if we can. It's not a showstopper. Certainly, you know, it may just impact hunger or well-being as we move forward. It's interesting as well that the potassium channel binding also showed up in this assay, and actually when you look at it's pretty similar to what you would expect. So this gives you about an 8.25 micromolar.
Are you going to get out to 50s at those? Because it'd be interesting to see what, if it's an agonist or antagonist, it could be serving whether or not the consequences of it.
Right. Yeah. And also, like one of the agonists, if it is an agonist, could be a positive allosteric modulator versus a direct displacement of the ligand.
Right. Yeah. No. I mean, that's we haven't scheduled that. I think it's something that we should do.
Follow-up is important.
Yeah. Yeah. Okay. I'll take that as a note for follow-up. So the dose escalation study is shown here at the bottom. So again, this was ran at Pharmaron, only looking at the oral dosing. So the 10 milligram per kilogram data was previously generated, and then we looked at 25, 50, and 100. And essentially what we see is the Cmax is essentially linear, is perfectly linear up to 50, and then it levels off a little bit as we get to the 100 mg per kilogram dose. But the AUC is essentially linear, so there might be a little bit of solubility issues that are leading to a slightly lower Cmax, but slower absorption and longer, a similar AUC. So, you know, I think this is really good. Good news. The MRT increases with dose, which, you know, again, might be related to that solubility issue. So we made around 5 grams of material. We can make more if it's necessary. We just completed a 5-day MTD study in these C57 black 6 mice. This is, as I said, we did this here. We looked at the doses here. We had a vehicle BID, 25 mg once and twice daily, 50 mg and 100 mg once daily. So all doses were well tolerated. We didn't see any deaths, body weight, or gross pathology, or behavioral issues in this study. The plasma was collected at 2 hours and 4 hours after the last dose, and the brains were collected 4 hours after the last dose. And that analysis is ongoing. The body weight change is shown here. So the vehicle group kind of gained body weight. The M4K 2009, all the groups kind of stayed around baseline, but certainly didn't lose weight. So that I think is good enough evidence to move the compound forward. And we're moving it into this FOP efficacy study with Dr. Eileen Shore at UPenn. The MTA is essentially complete, as I mentioned earlier. We're just waiting on signatures, and then we'll begin the planning study for that, planning of that study. We want to look obviously at efficacy and, you know, whether or not we can prevent aberrant bone growth, but also looking at dose response, looking at the PK/PD with respect to phosphosinide and ID1 expression, and then, you know, but we have to work with Eileen to really design that study.
You know how long it runs or how many days they dose?
It's about a month.
Oh, okay.
It's about a month.
So the FOP?
Yeah. Yeah. So it's basically it's got a 206H knock-in, and then they injure either the Achilles tendon, and you know, and then they look at bone growth in response to that injury. So so yeah, it should be quite interesting. I'm hoping Eileen is actually going to be able to present the model at one of the upcoming meetings, so that we can really get the expert. And then brain exposure. So we will get an initial read on the brain exposure from our MTD study, but we'll, you know, get real brain exposure at these higher doses at Pharmaron. Javad and his team at Children's National have the access to the real-time brain penetration of these compounds, and they have our compounds. So hopefully we'll get an update from them in the not-too-distant future as well. And then, you know, moving into the DIPG efficacy study from Chris and Engel and Jerome, I think have shown that, you know, there are several models that are available, and, you know, hopefully we'll be able to show some efficacy in those models as well. Target product profile, I'm running out of time, so I won't go through that, but M4K2009 looks really good. It's meeting the target product profile that we're targeting. Key areas to address are trying to continue to improve the potency and selectivity, certainly against ALK4 and ALK5, and David showed nicely that we've got some, I think, interesting paths forward there that are leading to enhanced selectivity. The hERG is something that, again, that we need to follow up. Is it really a liability? But, you know, we have some plans, some SAR plans designed to try to remove that hERG liability, and so we'll see whether or not they pan out. Potential off-target pharmacology identified in syrup, and again, we'll follow up with some assays there, and then the high brain tissue binding, and, you know, something that if we can improve, that'd be great.
I mean, it's a good thing for diffuse. That's what we were talking about. For diffuse, the DIPG, maybe it's not a bad thing to have.
High tissue binding.
Maybe.
Yeah, I mean, again, most of that data with the free fraction in the brain has been generated from things like GPCRs and other targets in the CNS. So we don't know whether or not that's really going to be the case with a kinase inhibitor. David really highlighted most of this data. I think we've got a couple of really interesting compounds that are starting to emerge. 2117, we actually have a great X-ray structure for now. It's more selective than 2009. It's more potent than 2009 in the nanograd assay, and again, more selective in the cell-based assay. Gives PK profile that's essentially identical, to that of M4K 2009. Maybe a slightly longer half-life, slightly lower AUC, but I think it's, given the variability that you see, I think it's really quite reasonable. 2118 is also a pretty interesting molecule. The pharmacophore here might potentially be a liability and might be something to study moving forward, but it's really nicely selective against ALK5, gives an even better clearance number and certainly a longer half-life here, so that's an interesting compound. 2143, this nitrile as well, is a very interesting compound that's potent and selective. It does have a little bit of efflux, and we've seen that can lead to problems in terms of brain penetration with one compound that we've looked at previously, but something to study. We have looked internally at the pharmacokinetics of this compound in our rapid screen, and it did show some reasonable exposure, so this is something that we will move forward into a full PK experiment. And I think some of the compounds that David highlighted in his presentation as well, I think are ones that will also move another subset of compounds into full PK experiments to try to generate that next list of compounds. I did want to say that the blueprint patent summary, so the blue 782, we think is this molecule, it's example 877 from their patent. We are going to have this compound made. We've outsourced it to one of our vendors that we have working with us on this project, and so we've actually requested 500 milligrams so that we're able to profile this analog as much as possible in our hands and look at how it impacts ALK2. They also ran this compound, so this is actually the surgery-induced model. This is very similar to the type of data that we'll generate, David. Shown here, you can see it actually, here they've got out to actually 50 days, it looks like. So, 50, okay. Yeah. And with that, I think, we continue to make great progress. I think M4K 2009 is continuing to move through the pipeline. I think we're going to be able to generate some very interesting data with that compound moving forward. And I think we continue to identify even better compounds. So overall, the project continues to be in good shape. And, I think getting M4K 2009 out to the community and trying to generate as much data as possible, looking at, longer proliferation assays, looking at any kind of DIPG in vivo assays that we can get the compound into, and certainly this FLP model, I think are all going to be very interesting data to help drive the project forward and try to define what we need and whether or not a compound such as this is going to work in DIPG. So, with that, again, I think, we went just slightly over, but, I think it has been 2 months since we had a meeting, and, we had some really great discussions. So, If there's any other questions or comments from anyone, then please speak up. Otherwise, then certainly we'll have a meeting next month in December, and I think we'll have a really great discussion then too. So thanks everyone for all the contributions. Thanks for all the contributions towards the paperwork and the reporting that happened, and let's keep pushing this thing forward. All right. Thanks everyone.
Thanks, Jeff. Bye.
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 Thank you. Bye.
Thanks, Alex. Bye now. Yes.